The last chromosomal aberration that we're going to talk about in terms of a structure change is a reciprocal translocation. This is simply a mutation event that happens within a cell, say a cell that's 2N for this, although obviously this can happen in haploids as well, where we have a breakage and rejoining such that these two switch places. Basically, this tip now becomes physically associated with this chromosome, and this tip of this chromosome is now stuck on the end of this chromosome. So what I'll do is I'll simply just draw that. So now this red chromosome has the tip of the black chromosome physically attached to it, and likewise over here, the red chromosome tip is now attached to the centromere of the black chromosome. Now what we'll do in the next little segment is we'll ask, well, what happens when this undergoes synapsis and meiosis? What does synapsis look like in an organism that is now a translocation heterozygote? And we'll tackle that in the next segment. As we've seen before with deletions and inversion heterozygotes, chromosomes are thin and flexible, so they maximize pairing to the full extent that they possibly can. In the case of a reciprocal translocation, we actually have 4 chromosomes that have at least partial homology with each other, such that they will all synapse together in what's called a cruciform structure. So let's draw what that looks like. So I always start by taking the 2 non-translocated chromosomes in the translocation heterozygote and drawing them at opposite corners. So these are the non-translocated homologs. Then we have a homolog that has this portion of the black chromosome and then the red tip. And then we also have the red centromere chromosome that now has the black tip. And if you draw them like this is the proper way to show maximal pairing between the 4 different chromosomes. So again, wherever there is pairing, there is synapsis, and wherever there is synapsis, crossing over is possible. So we have synapsis everywhere in this cruciform structure, except right here in the very middle. But this area is very, very small, again, because these are very thin and flexible structures, so they maximize their pairing. So here we have a translocation heterozygote. Okay, it's at synapsis. And we've got maximal pairing. Now it's time to think about what's going to happen genetically when this organism attempts to take this mess, as it were, and take this through meiosis, meiosis 1 and meiosis 2. The first thing to notice is that segregation patterns are now important. Okay, so segregation can now produce one of a few different things. Now, what we're going to do is we'll introduce some terminology here, and we'll actually start labeling centromeres. If I call the black chromosome chromosome 1, then I'm going to call that centromere N1. The translocated version of chromosome 1 I'm going to call T1. Again, based on the centromere. So I'll just give myself a bit more space in my centromere and call that T1. That would make this T2, the translocated version of chromosome 2, if we're going to call the red chromosome chromosome 2. Give myself a bit more space there. And then we'll have a normal 2, or N2, on this side of the equation as well. Now, if you think back to our discussion of independent assortment early on in the course, you'll remember that there are 2 possible segregation patterns. We can have this segregation pattern, or 50% of the time, independent assortment will take the opposite centromeres. When this cell undergoes meiosis, it doesn't know that it's dealing with a translocated structure. It simply wants 1 each of centromere 1 and 1 each of centromere 2, but it doesn't necessarily know what's attached to it. So as a result, we can have a number of different segregation patterns. The first one is we could have N1 and T2 going together, and then T1 and N2 going together. So N1 with T2 is that set going for up to one cell for meiosis 1, and the T1 and N2 going down for that first cell division at meiosis 1. The first thing you should notice is that these structures are not compatible with one another. N1 with T2 has a full chromosome 1 set of genetic information, but doesn't have a full set of chromosome 2. It's missing this whole region of chromosome 2. Also, it's duplicated for this tip of chromosome 1, so it has both a deletion of a region and a duplication of a region. And as a result, this is not a proper haploid set, and as a result, it will not form a functional product of meiosis. Likewise, the T1 and N2 segregation down here have duplications and deletions as well. The other segregation pattern that we'll see 50% of the time, however, is normal 1 and normal 2 on the one side, and translocated 1 and translocated 2 on the other side. And these will both work, whereas these did not work. And the reason for that is a normal 1 with a normal 2 and a translocated 1 and a translocated 2, these both have proper haploid sets. They have the right amount of genetic material from chromosome 1 and the right amount of material from chromosome 2, either in the normal structures or in the translocated structures. But there's no duplications or deletions in either. So that's just simply based on segregation. And we would notice that 50% of the time the products of meiosis that result will not be functional in this case. In the next segment, we'll go on to start talking about crossovers within these structures, which obviously makes things a bit more complicated. I've now added some loci to assist us in examining the results of different segregation events and crossing over within a reciprocal translocation cruciform structure at synapsis. So again, this is synapsis. And synapsis means that crossing over is possible wherever synapsis is taking place. And we have the meiosis 1 division as the first division that we need to deal with. Okay, this organism is also a dihybrid, so it's a translocation heterozygote, and it's also genetically heterozygous for 2 loci. And these loci are on separate chromosomes. So in the normal chromosome structure, we have the A locus on the one chromosome and the B locus on another. Now, perhaps in a question that you might deal with, you might have originally mapped A and B to be on separate chromosomes. Now we'll see what happens when a reciprocal translocation swaps the tips between these chromosomes, and now we have a big A and a big B on translocated chromosomes, and it's an organism that is a translocation heterozygote with little a and little b on the normal chromosome structures. The first thing to notice is that in the absence of crossing over, without any crossing over going on, the only outputs that we'll get are the little a and the little b together, and the big A and the big B together, if there's no crossing over going on. And the reason for that is, without crossing over, those are the only compatible structures. Normal 1 and normal 2 can work together. T1 and T2 can work together. And as a result, we would only get these outputs. We wouldn't see the recombinants. Now that's unusual. We expected them to be on separate chromosomes, and we expected them to produce recombinants at equal frequencies as the non-recombinants. We expected 1/4 for everything. Except now, recombination is dependent on crossing over. So what this does is it gives us one of our first diagnostics for a reciprocal translocation, and that is we have apparent linkage for unlinked loci. So things that we know are unlinked suddenly are behaving as if they are linked, and the reason for that is now it's going to take a single crossover in one of these regions to actually produce recombinant genotypes, because we need a crossover to start swapping chromosome structures to give us new possibilities for outputs. The inputs, of course, here would be big A and big B coming in from one, and little a and little b coming in from the other. And the reason for that is these are, again, the chromosome compatible structures. Little a and little b had to come in as a set because that's the compatible structure. Big A and big B had to come in as a translocated set because that's a compatible structure. You can't have inputs on, say, like an N1 and a T2. We couldn't have that as an input because that organism wouldn't be able to produce that as a functional product of meiosis because it wouldn't have a proper haploid set. Okay, so in the absence of crossing over, we're only going to get inputs. Now let's start asking, well, what happens if we do have crossing over taking place? And again, synapsis is here, so crossing over is a possibility. Okay, if you have a crossover, say in this region here, what that actually does is now gives us two dissimilar chromosome structures attached to the same centromere. And I should leave myself enough room to draw meiosis II, so let's just tighten this up a bit. Okay, so let's start tracing. This top chromatid was not involved with the crossover, so it has our little a on it and a normal chromosome structure. This chromatid, however, picks up the little a and then crosses over to give us the translocated structure, which we could draw like this. So if this is segregating up, this is what comes along for the ride. We now have a little a that is present on a normal structure, as we did before, but now the new thing is that we have a little a on a translocated structure. We haven't had that before. Now, this could segregate successfully with the other translocated structure. So if we take this one and put it up, we can draw that out, and that's just a standard T2. Okay, and it has big B alleles on it. So if that's that one cell after meiosis I, the cell down here, we should sketch that out as well. That would have now a normal structure with a big A allele, and that's different as well. We haven't seen that before. As well as what we had previously, the big A now with the translocated structure. Okay. And then down here we had just the regular normal chromosome 2 with little b's on it. So that's the other cell after meiosis I. Okay. Then meiosis II is simply, as before, segregating each of these into separate cells in order to give us the 4 products of meiosis. So some things to notice here. We now have a little a that is paired up successfully with a big B. So this is actually a way to get a little a, big B recombinant that survives, because now we have compatible chromosome structures. It's also a way to get a big A compatible with a little b, because now we have this big A allele which is crossed over onto a wild-type chromosome structure. So we would get this one here would be a big A with a little b that survives. Now, on the other hand, this big A with this little b is not going to survive. And likewise up here, the little a on the normal chromosome structure is not going to survive with this translocated structure. So we would have a little a, big B that is not working, and down here we would have a big A, little b that is not working. But the key thing is that we would have these recombinants that are now functional and products of meiosis. So a single crossover event here can start to give us combinations that are surviving that are recombinant. And this is what gives us apparent linkage for these unlinked loci, because now in order to get these recombinants surviving, we need a single crossover event here.